Hi everyone. Today we'll be talking about RNA sequencing and differential gene expression analysis. Now, next generation sequencing has been around for over a few decades now, and its impact has not only been seen in typical research scenarios, but has also been applied in countless clinical settings over the past few years. And so, in today's discussion, we will be looking about the various considerations that are necessary to conduct a robust differential gene expression analysis from your own RNA sequencing dataset. With that, let's get started. To fully understand the concept behind differential gene expression analysis, it is most prudent to start with a very solid example. And so, for our discussion, let's take advantage of these two guys over here and let's name them Popo and Chichi over here. All right. So let's assume for the sake of discussion that Popo and Chichi here all belong to the same species, and they're members of that species. And as you can see from these two examples, Popo has curly hair, whereas Chichi has spiky hair. Now, if we are the researcher, we are the principal investigator, and we want to understand why Popo has curly hair, which, for example, in this species is the dominant or the wild type phenotype, whereas Chichi over here has spiky hair, which is a unique trait for Chichi. And so if you want to know why they have differences in the hair structure, one can try to use various lines of evidences as embodied in the central dogma of molecular biology, right? So we can use either the DNA, the protein, or the intermediate between those two, which is the RNA. And for today's discussion, because we will be looking at RNA sequencing and differential gene expression, we will focus our attention on RNA. But as I will discuss at the end of this discussion, there are various caveats into using RNA as your information source. So going back to this example, what we can do is obtain RNA from Popo and RNA from Chichi. And we can submit these RNA samples to a RNA sequencing facility that will give us the sequence data for these two organisms. And let's say for the benefit of this example, we have 5 more variants of Popo. That means we have 5 more that have curly hair, and we have 5 more variants of Chichi here. That means 5 more samples that have spiky hair. And so we take the RNA, and that's what we will use for RNA sequencing and differential gene expression. Now, before proceeding any further, it is very important to highlight the reason why we do or why we prefer RNA sequencing over the more conventional assays such as qPCR. Now, in qPCR, we do the testing of genes one by one. That is, if we have a known gene, say for example, want5A, we test it, we design primers for want5A and we test it, and we can have a quantification between Chichi over here and Popo over here. And from that, we can say that indeed want5A is overexpressed, underexpressed, or no difference between these two samples. Now, of course, the problem with that approach that is quite obvious is that you have to know what these genes are. And if you don't have any prior hypothesis going forward, it becomes hard to specify or to target what mRNA species you are going to amplify and quantify. And this is where the advantage of RNA sequencing becomes apparent, because RNA sequencing amplifies and sequences all of the RNA species available in your RNA sample. And because of this, you don't have to have a prior hypothesis going into your analysis, going into your differential gene expression analysis. You can have a non-hypothesis driven approach in your methodology, and that allows a lot of flexibility into the direction as to which you could move forward into your study design. And so that's the major advantage that RNA sequencing has over qPCR. But that is not to say that we don't do qPCR anymore, because we now have RNA-seq. The fact of the matter is, after we have isolated and identified target genes from our RNA-seq, we still have to do qPCR or other assays such as fluorescence in situ hybridization to actually verify if these genes are actually differentially expressed between conditions. Now, going back to our friend here, Popo, let's assume that he is a eukaryote, and because of this, transcription occurs in the nucleus, whereas translation will occur or is expected to occur in the cytoplasm. And thus, when we extract the RNA, we are actually extracting both the nuclear and the cytosolic RNA present in this organism. And again, as we mentioned a while ago, most of the RNA that will be enriched in RNA-seq studies are mature RNAs that have the poly-A tail. We do this enrichment by adding into the RNA lysate what we call oligo-DT beads, which are essentially minute magnetic beads that have polythymine bound to its surface. And what these polythymine oligonucleotides do is to ligate with the poly-A tail in mature RNA that has undergone the process of splicing and polyadenylation. And we do this preferential selection of poly-A rich mRNAs because these poly-A rich mRNAs are what we normally associate with mature mRNA species, and those mRNA that are actually ready for translation, and therefore have higher correlation with gene expression rather than immature RNA that has not yet undergone splicing and polyadenylation. Now, once we have enriched for our target mRNA species, next generation sequencing requires that we fragment the sample either by sonication, enzymatic digestion, or other means. We do this because the quality of the base calls has been shown to decrease as the fragment size increases. And so next generation sequencing library preparation kits would actually require us to generate fragments that are within the range of roughly 50 to 300 nucleotides. You may also get further clarifications on this topic by looking at Illumina or other companies that manufacture these kits. These fragments will then be converted to complementary DNA or cDNA to convert the uracil nucleotides into thymine counterparts in the DNA. Adapter sequences will then be ligated into these cDNA fragments, which actually serves two purposes. One, it carries within it the primer sequence, which will kickstart the sequencing process. And also, it carries the indexing information, which is critical when sequencing more than one sample at a time. Finally, we are at the sequencing component of the workflow. And here we have two choices between a single-end sequencing and paired-end sequencing, wherein single-end sequencing simply means that we sequence from either the forward direction or the reverse direction. And paired-end sequencing, on the other hand, from the name itself, would mean that we sequence from both the forward and the reverse direction. The question is, why do we have to choose between these two platforms? The answer to that is actually twofold. One being that single-end sequencing is actually cheaper than paired-end sequencing. Although paired-end sequencing actually have higher probability or higher chance to find reads that span splice junctions. Now remember, only mature RNA have the introns taken out, and therefore reads that span these exon junctions are very informative and would mean that these reads are actually aligning to mRNA rather than contaminating genomic DNA. For single-end sequencing, we do not have this information, and most of the time we have these reads, single-end reads, mapping to actually either a single exon or to mostly exons only. And therefore, we cannot verify if we are aligning to or we are amplifying genomic DNA that is contaminants or the actual target, which is messenger RNA. So going back to our friends here, Popo and Chichi, the entire RNA-seq workflow would look something like this. First, we extract the total RNA from the scalp tissue we obtain from our samples. And next, we subject this total RNA to cDNA library preparation. And lastly, we do the sequencing. And from that, we get our sequencing reads. And that sums up the entire wet laboratory considerations and the principles going towards RNA-seq analysis. Once we get our sequencing reads from the sequencing facility, we cannot immediately jump into analyzing it using software. And we actually need to do a lot of pre-processing and alignment in order for us to arrive at the necessary input that is required by the differential gene expression analysis. And by this input, we mean the raw gene counts. And we will define what raw gene counts are later in this discussion. And for now, we have to focus ourselves on what are the necessary considerations for us to properly process our RNA sequencing data for the subsequent analysis. Here we see a typical workflow arising from a RNA sequencing experiment. And as you can see, there's a lot of work that has to be done prior to gene expression analysis. First is that we have to check for quality metrics and to trim for poor base calls. Next, we have to map our reads, our fragments, into a reference genome if the reference genome is available, or to map it to a de novo assembled transcriptome or genome. Lastly, we have to finally count the reads that actually align to the reference. What do we actually get from a RNA sequencing facility after we submitted our sample for sequencing? Here we see a typical output that a RNA sequencing facility will send out to their customers after the RNA sequencing has been finished. So you will see the actual main output, which is the FASTQ files. And these FASTQ files are sometimes compressed into the GZ compression format, or may sometimes not be compressed and just exist as FASTQ. And usually you don't decompress these files because they are already too big, and the softwares that you use to process this actually understands the FASTQ.gz format and will decompress these files for you on the fly, so you don't have to do anything. Down here, you will also see that we have some metadata for the actual sequencing that was carried out. This metadata would usually comprise of the sample names, the total yields that was obtained in base pairs, and also the percent of reads that have greater than or equal to quality 30, which is the threshold for next generation sequencing, and also the mean quality scores. If we inspect the FASTQ files, we will see that it contains a series of characters that on first look doesn't mean anything. But this actually contains the sequence information, the index, and also the quality for the sequence base calls that was carried out. So let's take a look a little bit more on what this FASTQ file actually tells us. So the first thing that the FASTQ file would show is the header. And the header would usually start with an @ symbol, followed by the information on the sequencing platform, the type of sequencing that was carried out, either it's single end or paired end, as well as the index sequence that was used. Following the header is the actual sequence, and as we mentioned a while ago, the actual fragment should be around 50 to 300 base pairs, followed immediately by the quality scores. Now, this quality scores is very interesting because it's not the usual number that we see from 1 to 30, but it is ASCII symbols. And these quality scores can actually be in Fred 33, Fred 64, Solexa 64, and others. But what we have to understand is that for NGS, the Fred quality score must be at least 30. And that means the probability of obtaining an incorrect base call is 1 in 1,000 bases. And that's a very good confidence for the base call. Lastly, we note that the FASTQ file actually contains all of the sequence reads from each sample that we have sequenced. And so this contains hundreds and thousands of sequencing reads. And what we are showing here is just the first 3 sequences of this file. And we expect more than 1 million reads from this sample. We also have to remember that when we request for paired end RNA sequencing, our FASTQ files will always occur in pairs. That is, we will receive a read 1 FASTQ file that is paired with a read 2 FASTQ file. And those 2 paired FASTQ files cannot be taken away from each other. They have to go with each other. Whereas in single end, you will get only read 1 sequences. And if the sequencing facility opt to separate this in separate lanes, you will find that it will show lane 1, lane 2, lane 3, lane 4, and so on and so forth, up to how many lanes the sequencing facility would use. And so this is a critical difference that you have to take note of when requesting RNA sequencing from a third-party facility. In bioinformatics, we have this very common adage or saying that goes garbage in, garbage out. And simply put, it means that if the initial file, initial input that we feed into our algorithms, into our softwares, is contaminated with poor quality samples, poor quality input, it would eventually lead to your output also being full of noise and being inadequate for downstream analysis and interpretation. And so we have to be very careful at analyzing the quality measures and quality metrics that we have for our files. And for this specific experiment, it would be the sequence files that we are interested in. There are a number of software that we can use to visualize and aggregate all of these quality measures that we need in order to fully process our FASTQ files. An example of this is the more popular FastQC software. And for further information on what features it has, I recommend that you go into their website and read their manual. But for this discussion, we will only look at the various metrics that FastQC would provide and explore on the metrics that you should be aware of and what the recommended values are for each. First among these metrics is the per base sequence quality. And for this, we have already mentioned previously that a good sequence quality should at least have a quality score of 30. Similarly, the per sequence quality should have a peak at quality 30, when we average the total quality score across the total number of reads that you have for a particular RNA-seq experiment. Next is the per base N content. And for this, it is actually not very unusual to find a low number of N's appearing in a particular sequence, especially when we look at the ends of the sequence. However, if this proportion increases over a set threshold, it might mean that the RNA-seq pipeline was not able to interpret the sequencing data enough to provide a valid base call. Another important metric in your FASTQ file is the adapter content. For FASTQC, it automatically determines the enrichment of specific and known adapter sequences that have been published by library preparation manufacturing companies. And so it would be easy to identify what adapters you need to trim from your sequence. However, if FASTQC does not detect any adapter sequence or provides ambiguous enrichment for adapters, it would be wise to contact your sequencing facility and try to get the adapter information from them so that you would be able to trim away these sequences from your reads. Do note that there are available softwares that would automatically detect these adapter sequences without prior information of what specific adapters were used during the sequencing. Once we have processed our reads, we are now ready to do the mapping of these reads to the corresponding reference genome. Now, do remember that mapping to the genome requires that the genome be as close as possible to the species that you are working on. For example, if you are working with human, you have to align to the human reference genome. And if you are working with dogs, you have to align to the equivalent dog reference genome. Aligning reads to the improper reference genome would yield very low mapped reads and would also invalidate all of the counts that you obtain downstream. Do remember that when using genome for reference during the mapping stage, that a splice-aware aligner is necessary. This is because the introns are present in the genome and would therefore prevent splice-unaware aligners to map properly to the reference. However, for reference transcriptomes, splice-aware and splice-unaware aligners would perform similarly. As a final note in mapping, it is critical to realize that differential expression actually requires less mapping accuracy compared with other genomic analysis, such as variant detection, allele-specific expression, genome annotation, and transcript discovery. And thus, if we check the accuracy for the popular aligners that we have nowadays, we would see that all of them would perform well enough for differential expression. Once you have finished with your mapping, it would be wise to assess the results arising from this step. Now, the critical information that you have to consider is the number of reads that map uniquely to a specific region in the genome. And by uniquely, we mean that it mapped to only one specific region in the genome. Whereas reads that map to more than one region in the genome, which is also termed as multi-mapped reads, should not be included in the final counting of reads. In addition, for paired end reads, remember that the forward and reverse sequences are matched. And so for paired reads that map to different regions in the genome, we also have to exclude those mappings. And so the critical threshold is not as clear cut, although you would want mapping results to be roughly 60 to 70% or more. Finally, as a last step before we do the actual differential gene expression analysis, we have to count the actual aligned reads in order for us to obtain the raw gene counts. But why does the manner of counting these genes actually matter that much? If we know the sequence of a specific gene and we also know the corresponding chromosomal location of this gene in the reference genome, wouldn't it be pretty straightforward to count these mapped reads? Well, unfortunately, for complex genomes such as in eukaryotes, the definition of a gene has actually drastically changed over the past few decades, ever since we have developed technologies such as the next generation sequencing. The discoveries of alternative start sites, the detection that we have alternative splice variants, and the fact that mRNA transcription can actually utilize both the forward and the reverse DNA strands as template has drastically complicated the counting process. On top of that, we also have the observation that unique genes can actually overlap in a genome. And so this clearly shows us the necessity of why we have to properly flag these mapped reads as either informative or ambiguous. In order for us to correctly assign a mapped read as being informative or ambiguous, we need to have a map of the reference genome to tell us which chromosomal locations actually correspond to unique gene features, such as exons, coding sequence, or stop codons. This map in bioinformatics is usually termed as the annotation file, which usually exists as either a .gtf file or a .gff file. And you can download these files in known genome repositories such as NCBI or UCSC. Now that we have our annotation file, we can proceed with filtering the map reads for alignments that have poor mapping quality, otherwise known as MapQ, and alignments that are ambiguous. let's inspect an actual mapping output file, which is either in .sam or .bam format. We can get a good impression of the alignment to the reference based on the first 5 columns of this file. The first column is simply the query name or the read name, which is usually assigned by the sequencing facility. Next is the alignment flags. Here we have a value of 83, which corresponds to a mapping with correct orientation and insert size, which means that this is an informative mapping. Next is the R name, or simply the reference name to which the read would align to. And this is followed by the POS, or the position of the leftmost mapping that aligns with the reference. And lastly, we have the mapping quality, or the MapQ value. Now, MapQ is actually computed as -10 times the log base 10 of the probability that a given mapping is wrong. So if we accept an error of 5%, for example, we should set the threshold for MapQ in our counting to be greater than 13. Understanding all of these mapping parameters is very important during the counting of the reads, or otherwise known as read quantification. Because during this stage, you should pay special attention to how you would treat multi-mapping reads. Would you wish to ignore those, or to actually still utilize the reads? In addition, you should also consider whether you would wish to count your reads at the transcript level or at the gene level. And you would also want to incorporate what threshold you wish to use for your mapping quality. Now, most would recommend a mapping quality of at least 10. And again, this depends on the amount of error that you would wish to accept in your experiment. Going back to our example for Popo and Chichi, we can therefore generate a table containing the counts of all the aligned reads based on our selected mapping quality, gene feature level, and handling of multi-mapping reads. Here we show an example of the counts matrix for the 5 RNA samples of Popo and 5 RNA samples of Chichi taken from their scalp tissue. The values that we see inside this table are the actual gene counts. These counts, which is usually denoted by the letter K for gene I of sample J, is proportional to the expression of that specific mRNA and is affected by factors such as gene length, sequencing depth, RNA enrichment, quality of RNA, library preparation conditions such as amplification and reverse transcription, as well as computational factors such as mapping and counting parameters, which emphasizes the need to be familiar with the various wet laboratory and in silico requirements for RNA sequencing data analysis preparation. A typical differential expression analysis would usually comprise of 3 major steps. First being the normalization of the raw counts, followed by the estimation of gene dispersion, and lastly, the statistical comparison of the estimates between groups. Once we have obtained the raw gene counts, we are now ready to conduct differential expression analysis between groups of samples. Over the past few years, a lot of tools have been developed to compare gene expression between conditions. Tools such as Lima, EdgeR, DSeq2, SumSeq, NoiSeq, CAFDIF2, and EBSeq. And the concordance of the results obtained from these tools are generally good, and most of the factors that would affect your choice of the differential expression tool that you want to use would mostly depend on the type of normalization that you want to use and the compatibility of that tool to other upstream and downstream tools that you would wish to utilize. In conducting the actual differential expression analysis, we have to pay attention to certain parameters that can significantly affect the results that we get, specifically the number of differentially expressed genes, or abbreviated as DEGs, that we get, and also the fold change values, which compares the expression of each gene relative to a reference. First among these factors is the experimental design. In most scenarios, a simple model including only one covariate would be more than sufficient for most use cases. However, batch effects and other covariates can also be added in order to account for the effects of these factors. Interactions between covariates can also be used for more complex analysis. Another factor is background noise arising from genes with very low but non-zero counts and genes that have high but non-significant expression between groups. Finally, one also has to consider whether to do log fold change shrinkage or not. The motivation behind log fold change shrinkage arises because of the fact that individual genes will tend to vary in terms of biological variation, such that in cases where we have low sample sizes, gene dispersion estimates tend to be very poor. In addition, the presence of genes that have low non-zero counts tends to misguide fold change estimation. Imagine two genes that has been deemed statistically significant, and let's name these genes A and B, such that the counts for gene A in the test group is 5 relative to its count in the reference group, which is 1, for example, leading to a fold change of 5 divided by 1, or just 5. In gene B, the counts ratio for test and reference group is, for example, 5,000 is to 1,000. And therefore, we compute the fold change as 5,000 divided by 1,000, which is also 5. Even though both of these genes have the same fold change, a simple inspection of the counts would reveal that only gene B is biologically significant and has a good chance of being validated by qPCR. Thus, the fold change shrinkage attempts to strongly moderate for genes with low counts, but weakly affect genes with high counts. And in doing so, provide a better visualization of differentially expressed genes. Once we have implemented our chosen analysis workflow, we can generate various tables and visualizations to aid in data interpretation. The first thing that you would obtain is usually the results file containing the estimates from the comparison of gene expression between groups. The key information here is the false discovery rate p-values, otherwise known as p-adjusted values, as well as the log2 fold change. In differential expression, genes can be filtered based on statistical significance, which is based on an adjusted p-value less than 0.05 or 0.01, and biological significance, which may differ depending on your preferences. Usually, we use an absolute log2 fold change of greater than 2 for most applications. Next is the normalized counts file. This counts file can be used by other visualization tools such as heat maps and volcano plots to get an initial impression of individual gene expression for selected genes. Before doing any interpretation and further analysis, however, always make sure to conduct exploratory data analysis by principal coordinate analysis or PCA to check for the agreement of gene expression within groups and the separation of samples among groups. Also inspect that the MA plot and dispersion plots do not show any unusual patterns such as high dispersions, which would affect your analysis. You can also plot a p-adjusted histogram to check if you have enough statistically significant genes in your data. Usually, you would expect roughly 2,000 genes or more to be statistically significant at this level of filtering. These differentially expressed genes usually cluster around specific pathways. And so you can also utilize gene set analysis tools to identify enriched pathways and check for overlap of overexpressed or underexpressed genes in your data with known gene sets in existing databases, such as KEGG, MSigDB, which contains the gene ontology features, Reactome, and other such databases. This analysis is also termed as gene set analysis or pathway analysis. And this would help you in identifying therapeutic targets and critical genes that can be manipulated for other functional studies that may arise from your initial bulk RNA-seq analysis. That was quite the journey, isn't it? From finding samples to extracting your RNA, to sending it to your sequencing facility, and in doing all of that work to pre-process your data, and finally to analyze it using differential gene expression algorithms and tools. Quite a long journey. And as I have promised early on in our discussion, here are some of the final thoughts that I have for RNA-seq for your research. First is that you have to understand that RNA expression is actually tissue specific. And what that means is that mRNA that you get, for example, in skin tissue, will have different levels of expression for different genes when you compare it with, for example, retina or eye tissue. And not only is it tissue specific, but it's also temporal specific. And that means different developmental stages would also give you different mRNA expression for these different gene species. And so that adds a lot of complexity into the timing that you have to consider when you are collecting your mRNA samples. Next, which is also related to this first factor that we have discussed, is that RNA integrity and RNA quality is very important when you are doing RNA-seq analysis. The fact of the matter that RNA degrades faster than DNA means that we have to be prepared to accommodate for more complicated extraction processes, especially for not so common tissue samples. And of course, because RNA is not as easily extracted from exotic tissue samples, for example, in live individuals, you also have to consider where you want to extract your RNA. And lastly, one of the most important things that you have to understand is that RNA expression does not always equate to protein expression. And so in certain instances, you would see that a certain gene would have a very high RNA expression, but when you validate in qPCR or in immunofluorescence, you would see that these proteins do not actually have very high abundance. And so one must pay extra attention to not be satisfied with RNA-seq analysis. That is to say that once you have your candidate DEGs or differentially expressed genes, you have to go to the extra step and validate this using other assays such as immunohistochemistry, fluorescence in situ hybridization, and some other assays that you would wish to do. And these are all the thoughts that I would want to share for this discussion. And I hope you would understand that the concept and the topic of RNA-seq is quite a big field, and it's always constantly evolving across the years following its conception during these last few decades. And so trying to cram all of this information into one video, one informative discussion might not be possible. But I hope you found some pieces of information that would help you in your own analysis and would enlighten you into the decision making that you would carry out in setting up not only your wet laboratory decisions, but also the algorithms and the parameters that you input during your differential expression analysis. And with that, I thank everyone who is listening to this discussion, and I hope to see you in the next one. Bye-bye.